Welcome to immunology. I'm so excited. I do love immunology. It's very interesting topic, and I'm so excited to start this journey through immunology. First, we're going to do a chapter overview. In this chapter, we will be mostly learning the language of immunology. My family members have medical backgrounds and law backgrounds, and they've all told me that the first thing that they have to do before they learn anything in their professions is learn to speak the language and learn the terms. So we'll be learning a lot of new terms that will be interesting. We're first going to be talking about immunity and a little bit on immunization. Toward the end of the course, we will discuss more about immunizations. We're going to cover innate immunity versus adaptive immunity, and so don't get overwhelmed already. We're going to define those terms. We're going to discuss the cells of the innate and adaptive immune systems, which ones are shared, which ones are different, and then the organs of the immune system. Immunology is the study of the reactions of a host with foreign substances introduced into the body. And your guided notes are printed for each lecture, and they go right along with the PowerPoints. I try to highlight the words that go in the blanks so you can follow along there and actively participate in lectures. Antigens and antibodies. Immunology is all about antigens and antibodies. And what we're doing in immunology is we're laying the foundation for blood banking that will be taught a year from now. And it's very important that you understand the difference and the relationship between antigens and antibodies. So the antigens are the foreign substances that induce a host response, and they are all around us in nature. And they could be so many different things, from pollen, bacteria, viruses, anything that's going to cause an immunity response. The antibody is the serum or plasma factor made in response to a foreign substance. So let's stop and define serum. So that's probably a new term. When we draw blood, we collect a tube of whole blood that has red cells and liquid and white cells and platelets. So depending on what kind of tube that you drew the blood in, the liquid portion, if it has coagulation factors in it, it is called plasma. And if the blood is allowed to clot first and then you centrifuge it and the clotting factors are all in the clot with the red cells, then you have serum left behind. So back in the day when I first learned about antigens and antibodies, the definition of an antigen was a substance that elicits an antibody response, and the definition of antibody was something that is elicited by an antigen stimulation. So that was not helpful. But we're going to dive into antigens and antibodies. Immunity is the condition of being resistant to infection, and that includes several different things, like as simple as inflammation, which is a non-specific immune response, phagocytosis, antibody synthesis, and complement. So antibody synthesis is very specific, and complement are a group of proteins that are initiated in certain immune events. So I told you that immunology is all about antigens and antibodies. So my favorite antigen is the picture of pollen under electron microscope that looks just like the Death Star. So in this case, if we were comparing pollen and the Death Star to an antigen, what would be our antibody? Of course, Luke Skywalker. So you get it. An antigen elicits an antibody response. Now we're going to talk about the different classes of immunity. So we're going to talk about innate immunity versus adaptive immunity. So the first one we're going to talk about in detail is innate. This is also known as natural immunity because this includes just the normal body functions that are present to resist infection. So that's things like skin, the pH of urine, We're going to talk about those in detail in a separate lecture. For innate immunity, you don't have to have any previous exposure, so it doesn't matter if you have the same cold or if you've seen it before or not, your body is just going to attack. This is considered non-adaptive or non-specific. The response is going to be the same for any pathogen or foreign substance that you're exposed to. And the response does not change with subsequent exposures. So that means it's going to be the same the first time you see that pathogen as the 10th time you see that pathogen. And I added this meme of Dory here that says, I'll email this to myself so I can remember it. Hey, look, a new email. And this is to drive home that innate immunity has no memory. It does not remember anything. It just is, it attacks and asks questions later. So when you're thinking about the innate immune system, think about Rambo or Predator or the first Terminator. The immunology is relatively a new subject, a new topic of area of study. So we're going to talk about just two people in the history of immunology. So Edward Jenner was the first to discover or notice that milkmaids who got cowpox did not get infected with smallpox. And so his work led to cross immunity. And so that means that the milkmaids had cowpox, which gave them immunity to a worse disease that was smallpox. So make sure that you can associate Edward Jenner and his work with cowpox and smallpox with cross immunity. Louis Pasteur, he worked with particularly rabies vaccinations, and he discovered that you could use an attenuated virus. So we need to define attenuation. And attenuation means that you've made a pathogen less virulent. That could take place through heating or aging or through chemical means. So you don't need to know the exact experiment, but Louis Pasteur used a cholera bacterium and chickens, and he came up with the idea of inducible immunity. So this is what led to vaccination. Phagocytosis, we're just going to go briefly through this because we're going to talk about it more in depth in a later lecture. But phagocytosis is part of the innate immune system, natural immunity. It consists of four main steps. There has to be physical contact between the white blood cell and a foreign particle. Then there's the formation of a phagosome, and then there's fusion with cytoplasmic granules to form a phagolysosome, and then digestion and release of that debris to the outside of the cell. And this is the cutest video of phagocytosis. So this is a video of a phagocyte, or some type of white blood cell that is diapedesing toward bacteria. It's going to catch it in a second, but this is what phagocytosis is all about. So there it caught it, it's engulfing it, and it's forming a phagolysosome, and it's going to digest it and put it right back out of the cell. Oh, that was so much fun. All right, so we need to talk about a couple of more terms. So opsonization is something that it means that phagocytosis has been enhanced. And opsonins are molecules that attach to a foreign substance and help prepare it for phagocytosis. Examples of opsonins include C-reactive protein, complement components, and antibodies. And C-reactive protein and complement, they are part of a huge group of chemicals called acute phase reactants. And these are just molecules, proteins that increase during things like inflammation or nonspecific diseases. and those are opsonins. So I like to think of opsonins as salt and pepper, because salt and pepper is something that we put on our food to make it more appealing. It sticks to our food, and then we want to eat it more. And also a salt with a deadly pepper, because I'm, an 80s girl. Just think about how a salt and pepper make foreign substances more appealing, so the white cells and immune system want to destroy it. So I hope that helps bring that point home. Okay, we've been talking about the innate immune system. Now we're going to describe the adaptive immune system. This is just the definition. So for adaptive immunity, this is a type of resistance characterized by specificity for each individual pathogen or microbial agent. So remember, this is opposite. This time in the adaptive immunity, it's very specific. You have the ability to remember a prior exposure. So I put this elephant with a memory stick and here in the bottom to help you remember that adaptive immunity does have a memory. And this results in an increased immune response. So it can boost your immune response. It's a little more potent than your innate immune system. So it's a specific for adaptive and also memory. Remember those. Okay, so now we're going to jump back over to cells of the innate immune system. So we're going to talk about cells of the innate immune system. So we're going to talk about leukocytes. And where are leukocytes, or white blood cells, WBCs, found? They are found in peripheral blood. That means the blood that is circulating through our veins and arteries. Now, since we're learning new terms and how to speak the language of medical laboratory professionals, I'm going to let you know a secret that we abbreviate peripheral blood as PB, kind of like peanut butter and jelly, but if you see PB, that means peripheral blood, not peanut butter. These leukocytes, the white blood cells, there are 5 types in our body. They play a key role in both the innate and adaptive immunity, some of them. So the 5 types are neutrophils, eosinophils, basophils, monocytes, and lymphocytes. And keep in mind that, we're fixing to talk about how there are 3 different types of lymphocytes: B cells, T cells, and natural killer lymphocytes. T cells and natural killer cells are part of the innate immune system. B lymphs are not, and we're going to explain why in just a minute. So neutrophils, eosinophils, and basophils are in a special group within the white blood cells. They are granulocytes because their cytoplasm contains granules, and those granules contain chemicals. Luckily, today we're not going to talk about the chemicals in those granules very much. Neutrophils. These are circulating in peripheral blood, and when we talk about the percentage, you're going to learn how in this program to count a white blood cell differential. And counting a white blood cell differential means that we look in the microscope and we count 100 white blood cells, and we number how many neutrophils, how many basophils, how many eosinophils. We count them exactly, 100, and then we stop. So within a 100 cell differential, normally you would count 50 to 75 white blood cells in adults. So that means you would see 50 to 75%. These neutrophils are multi-lobed, that with the nucleus. We call them segmented. Their granules stain neutral, and their main function is phagocytosis. So on these slides, on the individual cells, you need to know about what size they are, what their nucleus looks like, what their granules look like, and what their main function is. So another term, I'll teach you the lingo. We call these segmented neutrophils, or segs. So if you hear us say seg this and seg that, we are talking about segmented neutrophils. Eosinophils they're probably my favorite white blood cell because they are so pretty. Eosinophils in the innate immune system, they make up 1 to 3% of white blood cells in non-allergic patients. So we're going to get to that in a minute, but what do you think that means? If 1 to 3 is normal in non-allergic patients, their granules stain orange or red. Their main functions are in allergy reactions and parasitic infections. So if you have an allergy or a parasite infection, you are going to see more eosinophils. Basophils, there are very few basophils normally circulating in our peripheral blood. The granules contain histamine and heparin and some other cell chemicals and growth factors. These granules stain blue, black, or purple. They're just very dark, and this is honestly not a great picture of a basophil, but we're talking about these granules right here, these little black dots, purple, blue, black dots. The main function of the basophil has to do with hypersensitivity reactions, and there's a whole chapter on hypersensitivity. So basophils are put into action if a person is allergic to peanuts or bee stings. So that's what we're talking about, a hypersensitivity reaction that can be very, very serious. Monocytes are the next type of white blood cell. They are the largest white blood cell in peripheral blood. The nucleus has a horseshoe shape, and if these cells are circulating in the bloodstream, they're called monocytes, but monocytes can migrate to tissues, and then we call them macrophages. So remember that monocytes in tissues are called macrophages. It's kind of like they went through a schooling process, and they have a little more knowledge now, so we call them something else. And macrophages all over the body can have a different name depending on what organ or what area of their body that they are in. For example, macrophages in the liver are called Kupffer cells, and you see that there spelled on the slide. Tissue cells, remember, monocytes in the tissue are called macrophages. They play a role in initiating and regulating innate and adaptive immune responses. So within the innate immune functions, what they have, they can kill bacteria, they have an anti-tumor activity, they can help eradicate parasites, they are important in phagocytosis, and they can secrete cell mediators. So macrophages, very important, and remember they are one of the cells that can perform phagocytosis. Mast cells resemble basophils, but they come from a different lineage, and we're going to talk about the family tree of red cells and white cells. We're going to talk about how they develop beginning from a stem cell, but not today. But that's just one of the things that you can look forward to learning. Mast cells play a role in allergic reactions. They can also function as antigen-presenting cells. They can also enhance and suppress the adaptive immune response, but they're pretty and have these dark blue-black granules all over the cytoplasm. Dendritic cells are a different lineage from the lymph system. They are covered with long membranous extensions that make them look like nerve cell dendrites, but they are different. These are considered the most effective antigen-presenting cell in the body and the most potent phagocytic cell. So remember this, dendritic cells and antigen-presenting and phagocytic, we're going to learn more about antigen-presenting in the future. We're just not going to get very detailed on this very first lecture. So to me, this dendrite, I can remember that it's a good antigen presenting cell because it just looks so scary, and it's the most potent phagocytic cell. So of course you can just see it going through the bloodstream, gobbling up everything. And I had to put this next picture in here of the Demogorgon from Stranger Things, because these two cells, the dendrite, and the Demogorgon just look so much alike. It's like they are ready to destroy anything. Okay, so you might be wondering, how do cells get from the bloodstream to the tissue? Well, diapedesis is movement of the cells through the blood vessel walls. And they do this by forming a little pseudopod. So this right here, this cell, this neutrophil, is squeezing through the space in between the epithelial cells with a pseudopod and performing diapedesis, so movement through blood vessel walls as cells exit vessels. Now, why do they do this? They are moving toward chemotaxins. Chemotaxins are chemical messengers which attract white blood cells to an area. And we're going to talk a lot more about what those chemotaxins can be and how they come about much later on in the course. But chemotaxis is the process by which white blood cells are attracted to an area by chemical messengers. So the terms chemotaxis and chemotaxin can be a little confusing, but make sure that you get those straight because I think there is a test question. Hint, hint. Okay, now we're going to talk about cells of the adaptive immune system. Remember, adaptive is memory and specific. The lymphocytes are the cells in the adaptive immune system. Lymphocytes normally represent between 20 and 40% of the circulating white blood cells, and there are three types: B cells, T cells, and natural killer cells. B cells, we're going to talk about them on the next slide. T cells recognize foreign antigens on cells bound to MHC molecules. Now, I know that's a lot, don't get overwhelmed, but we're going to talk about how our cells in our bodies have these markers on them called MHC molecules, and that's the marker that lets our immune system that our cells belong in our body, kind of like it's planted a little American flag on every cell in your body so that your immune system knows, hey, I belong here, don't bother me. Natural killer cells, they are just what they sound like. Think Rambo, Terminator, Predator, any bad character that just goes around killing people, asking questions later, or killing things. Not that I'm promoting violence, but natural killer cells target tumor cells or infected cells without prior exposure. They just see a problem and they terminate that cell. Plasma cells are B lymphs that are actively secreting antibody. So remember, plasma cells, B lymphs, a core memory. And I put this here for my youngest daughter. Bless her. She's done a lot of things as a little kid, and she always says that we remember those. So there you go. Plasma cells, memory, B lymphs grow up to become plasma cells that secrete antibodies. And the plasma cell is so interesting even to just look at. You have the nice round nucleus here, and then think about what's happening. Antibodies are proteins, and so what we see here is the endoplasmic reticulum, or the endoplasmic reticulum, where you have all the ribosomes and machinery packaging proteins. So I like to think of a plasma cell as the Amazon shipping warehouse during Christmas time. I imagine they employ more people, they expand the warehouses, they're sending out more packages, and that's what happens in our B cells. It starts to make antibodies, so it's a plasma cell, and it has to expand its equipment to make all those protein parts. All right, now we're going to talk about the organs of the immune system. We have the primary lymphoid organs, and there are only two of them: the bone marrow and thymus. Those are the primary lymphoid organs. The bone marrow is one of the largest tissues in the body. It fills all the core of the long, flat bones, and it's the main source of hematopoietic stem cells. Now, hematopoietic stem cells, that sounds like a mouthful, but those are just the stem cells that give rise to all the erythrocytes, which are red blood cells, all the granulocytes, which we know now are neutrophils, eosinophils, and basophils, monocytes, platelets, and lymphocytes. And we're going to learn later on in this course and others, we're going to learn about that family tree that starts with the hematopoietic stem cell. It's very interesting. The thymus is a small, flat, bilobed organ found in the throat, And this is where T cells go to mature. T lymphs and T cells, that's the same thing. I use those interchangeably. And thank goodness, this is our first memory clue. Thymus is where T cells mature. So at least T and thymus matches. You're going to remember that. So primary lymphoid organs, there are only two: bone marrow, thymus. The secondary lymphoid organs, they function as sites for contact with foreign antigens. So think about it. We need a lot of places where we can come into contact with our foreign antigens that might be trying to attack us to make us sick. These secondary lymphoid organs increase the probability of immune response, and they include the spleen, which is really just a huge filter, the lymph nodes, which is also, they're little smaller filters placed throughout your body, the appendix, the tonsils, and the mucosal associated lymph tissue. And this is in your esophagus, your respiratory tract, your GI tract, and also then the cutaneous associated lymphoid tissue. The spleen, in particular, its job is to destroy old red blood cells. You're going to learn in your courses that the red blood cell has a lifespan of 120 days. So what happens at the end of that 3 months when a red blood cell dies? It is filtered out by the spleen, and some of it's even recycled. I know, spoiler alert. It also filters infectious agents and foreign matter. So the job of the spleen is to destroy red blood cells and filter the bad stuff that's coming into our body. The lymph nodes are the central collecting points for lymph fluid. It's where the specific immune response to foreign antigen occurs, and we don't really care about the secondary follicles. But lymph nodes, they're scattered all throughout the body, and this is where antigens come into contact with our immune cells mostly. Mucosal associated lymphoid tissue is found on mucosal surfaces like the GI tract, the respiratory and urogenital tract. It's a main port of entry for foreign antigens, and it has a lot of macrophages and lymphocytes. And we're also going to learn about some antibodies that are there. Another spoiler alert. It's so exciting. Okay, so in summary, the innate immune system is the ability of the body to resist infection through means of normally present, nonspecific body functions. Adaptive immunity is characterized by specificity, memory, and dependence upon lymphocytes. All blood cells arise from hematopoietic stem cells in the bone marrow. There are 5 principal types of leukocytes, and they are neutrophils, eosinophils, basophils, monocytes, and lymphocytes. Tissue cells involved in immunity include mast cells, dendritic cells, and macrophages. Cells that are involved in the innate immune response and are actively phagocytic include neutrophils, monocytes, macrophages, and dendritic cells. Lymphocytes are the key cells involved in the adaptive immune response. B cells are a type of lymphocyte that develop in the bone marrow and are capable of secreting antibody when mature, which means plasma cell. And T cells acquire their specificity in the thymus and consist of 2 subtypes, CD4 and CD8, and that will be important later on in the course. Natural killer cells they kill virally infected or cancerous target cells without previous exposure. The bone marrow and thymus are the primary lymphoid organs, and the spleen, lymph nodes, MALT, and CALT are the secondary lymphoid organs. And that concludes Lecture 1 of Immunology and Serology.